Today, our presentation is focusing on a new way to engineer an adenohypophysis on chip. So the first question which stands up is, what is an organoid? So, as you may know, an organoid is a 3D multicellular structure which tends to reproduce the microanatomy of a real organ. So it may exist different kinds of organoid. So you can find lungs organoids, skin organoids, retina, and even tumoral organoids. So this field of research dramatically increased since the last 10 years, as you can see on the graph following on the slide. And the organoids really brought a real interest, and there is 4 main applications of these interests that I will quote. So the first one is the modelization of disease. So we can modelize genetic diseases, cancer diseases, but we can also make drug screening from organoids, so we can analyze the interactions between molecules and the different tissue of the organoid created. But we can also use these organoids in regenerative medicine. Just we can obtain organoids derived from patients, and we can use the IPS to restructure some tissue and then transplanted in the patients to restore the biological functions. And finally, we can use organoids to study development of an organ of interest in animals. So the organoids also bring solutions to reduce animal experiments, since you all know that animals are still very often used in research. So as far as we are concerned, our team has been selected for the International Organoid Contest. So what is the aim of this contest? This contest is open to all students coming from master and engineer level. So the aim is just to select an organoid of interest to enter in collaboration with a reputed laboratory, and then each team can just hoping to get 3 medals. So the first one is a bronze medal, so which corresponds to a single cell type organoid. The second one is a silver medal, which corresponds to the multicellular organoid, and the last one is the gold medal, which awards the team which enter in connection, in collaboration, and connected their organoids. So, the final symposium took place 2nd of December of this year, and it was at the Sorbonne University. So let's focus in the subject. So the adenohypophysis. The adenohypophysis, as you all know, is the anterior lobe of the hypophysis. The hypophysis is a tiny gland located in the center of the brain and is responsible of the release of the 6 main hormones in the body, as for instance the growth hormone, the stress hormone. And once this gland is affected by some tumors or other diseases, it can involve perturbation of the secretion of those hormones. And as for instance, some tumors can develop on the hypophysis and compress healthy tissue and just prevent the release of hormones. There can also be hypopituitarism or hyperpituitarism. And this is why it is crucial to just get an adenohypophysis organoid to bring some new solution and therapeutic solution. So the first step of our research was to just mimic the in vivo development. So just to briefly describe the development, you can see that the embryogenesis of the adenohypophysis takes place between the day 13 and 51. And as you can see in the first images, there is two layers, one in yellow, which is the ectoblast of the stomateum, and the other one in blue, which is the infundibulum. And those two layers tend to enter in contact and just to form a vesicle, yellow vesicle, which correspond to the Raskot patch. So how did we manage to do it in vitro? So we didn't go beyond the Raskot patch formation because of a lack of time for the realization of our experiments. But we validate step by step the realization by looking and observing the presence or not of some markers, so the quotin markers, SOX2, PAX6, NKX2.1, PITCH1, and LH63. So it was really challenging for us because there is few articles which are dedicated in the subject, but we focused on the last one, so below, the article of Ozone, because he was focusing on 3-day culture, so the culture of interest to realize organoids. And we tested first two different medium, so the E6 and the GFCDM, because we didn't know which one was more likely to produce the organoids. And we tested those medium on 3 different IPS lines coming from patients from imaging institutes. The different factors that we used to create the adenohypophysis was the 4 that are there that you can see. And so from day 1 to day 6, we use a Rho kinase inhibitor just in order to create the aggregate, the embryoid bodies. And after, from day 6, we use the BMP4 for the co-induction of the oral ectoderm, which correspond to the outer layer of the organoid, and the hypothalamic neuroectoderms, which correspond to the inner layer. Then we use SAG and FGF2 treatments, which involves the induction of the pituitary placodes, and finally the invagination of the Raskot patch and the vesicle. So this is a description of our protocol. So as you can see on the 96 well plates, the place is divided by 2 sides. So each side for each medium. And this is composed by 4 different steps entangled together. And the first one is the creation of the embryoid bodies. The step 2 is the co-induction of oral ectoderm and hypothalamic neuroectoderm with the BMP4. The step 3, the cranial placodes, and the step 4, finally, the creation of the vesicle. So here are just some representative pictures of the development of the adenohypophysis. So as you can see, the size of the organoid is increasing in function of time, and especially since the moment we had the growth factor and the differentiation factors at day 6. But we had to stop the number 5 cell line because in the well we didn't observe a well-structured organoid. We just observed organoids which tends to disaggregate. So that's why we stopped the line. And finally, concerning the results, we made some immunostaining and PCR. On the figure 1, you can see that immunostaining of the number 4 cell line shows some co-expression of NKM and SOX2. So with this co-expression, we validate the neural path of the organoids along all the formation during the 27 days. And then we focused on a PCR on the 2 lines of interest because we stopped the number 5 lines, as I said. And we decided to focus the last immunostaining on the line 4 because we saw that the transcript NKX2.1 and LH63 were really expressed in this line. And the last immunostaining allowed us to validate the induction of hypothalamic neuroectoderm and pituitary placodes.
So during our experiment and by reviewing the literature, we observed that there was a lot of issues with the current organoid cultures. There was some abnormal structures that tends to emerge, and the big issue is that there was a lack of homogeneity between the samples and between the protocols. And there was also a lot of internal cells over the time. And another issue is that culturing organoids and cells in well-placed or petri dish is in static environment, which is very different from the dynamical environment of the body. So our objective was to use a new technology to culture organoid, which is microfluidics, to first optimize the regularity and reproducibility of the protocol, to miniaturize the culture, and by that saving cell mediums and factors, which are quite expensive when we perform cell cultures, but also to atomize the cell culture processes, and also to better reproduce the microenvironment which the cells are cultured in. So for this experiment, we use a very specific microfluidic device, which was developed by Charles Bauer and his team in Pasteur Institute, which is a microfluidic droplet array with anchoring system, which is represented here. We have a top view of the system. So the cells are loaded in the droplet of water, which is bathed into a null medium, and the cells are trapped into the red part of the trap, and we can add later small molecules such as antibody or growth factors, which are anchored by the blue anchors here, and are then trapped into the trap with the organoid. So as to validate our macrofluidic model, we use a simpler and quicker organic model, which is the retinol model, because it is because we can see the first structure of differentiation in day 20. So we use mouse embryonic stem cells with lineage D3. We use 3,000 cells per well in a 96 well plate and 600 cells per trap in a microfluidic chip. We use a very specific medium to induce the differentiation, which was composed mainly of Glasgow medium and knockout serum. We use half of the medium on the first day of culture, and after 1 day, when we begin to see the aggregations of the cell to form the embryoid bodies, we added 2% of matrigel, and on day 4, we added the other half of the medium, which was N2 supplemented. So our chip was contained 6 traps, 6 hexagonal traps of 6 microliters volume, and the cells were okay. So there was supposed to be a video here, but anyway, so the cells were supposed to be are loaded into this trap with the droplets. And here, after 1 day of culture, we can see the aggregation of the cells in the center of the trap and the formation of the embryoid body, which is the first step to obtain an organoid. Never mind. So as our preliminary result, we were able to observe the co-expression of RAX and PAC6, which are retinal pigment epithelium markers on day 14 of culture. And we also observed the expression of rhodopsin in blue, which suggests a differentiation oriented toward a retinal organoid.
So in order to create our 2 organoids, we had to consider our work as a full entrepreneurship. And so here is the composition of the team. So from the left to the right, there is Camille Brouillon, Guillaume Mondon, and Nessim Richard. So as you can read, we had just benefits from our different backgrounds, so different skills, and so we just had complementary skills. Concerning the project timeline, first we began on February of this year by bibliographic research on the field of organoid. We have, beginning March, a real and clear idea of what we wanted to do and the project to build. And we created the 1st of April our association named Combines. Finally, we met numerous laboratories. We explained our project to various researchers, and we were finally hosted by 2 major labs, Pasteur Institute and Imagine Institute. So we started then a large communication campaign just to access money and finally identify sponsors and financial partnership to finally start the experiments from June to September. Regarding our budget plan, it was estimated at 14,000 euros. And as you can see, we focused this estimation on 2 articles. So ozone for the retina and ozone for the adenohypophysis, and Equipe for the retina. And we benefit of help of our university, elevated at 1,500 euros.
So to conduct our project, we analyzed the strengths and the weaknesses of such a project. So using a SWOT table. So our main strength was that it was a very innovating project in a new field of research with promising medical and research perspective. But we had limited skills and expertise in cell cultures and in the field of organoid because we were only M1 students. We didn't have a laboratory to host our experiment, and we didn't not have a lot of money to start the experiments. We only have 10% of our estimated budget. So we focused our work in 3 axes: the scientific networking to find some laboratories to host our experiments, but also to find supervisors which were experts in the field of organoids and microfluidics. And we worked a lot on the communication to be able to raise funds and to find sponsors. So as Julie said, we met a lot of researchers in the field of developmental cellular and molecular biology, also in genetics and in stem cell cultures and microfluidics, who we presented our project to and who assessed its experimental feasibility. And we were able to find our host laboratories and supervisors, Dr. Nathalie Lefort for the hypophysis organoid and Dr. Charles Barou and Sébastien Sarr for the microfluidic experiment. So we build a physical communication through flyers that we use to approach and promote our project among sponsors during conferences and symposium, and also with a poster. We created a website to present our project, which you can find on the link below. And we also did 2 interviews, one in a YouTube channel, BTLV, and one in Capital Santé. And we also had an article in a CNews newspaper.
So as you all understood, the organoids offers really high perspectives. So amongst all those perspectives, we could Imagine Institute suggest us to have some collaboration with Tree Frog Therapeutics. So Tree Frog Therapeutics just created a device. This device is involved in the encapsulation of stem cells with a shell of alginate. And this shell allows the stem cell just to aggregate, to grow, and finally to differentiate, and allows the flow of nutrients through the alginate. So you can see below some images obtained with Imagine Institute of aggregates of adenohypophyses in shells.
So a main idea of the INOC contest is to promote the collaboration between teams of researchers and students. So as part of the competition, we did a collaboration with Firendo from Strasbourg. We helped them build a model of organoid solidity with respect to cell lineage, medium, and incubation time, with the formula that you can find here, with Y being the solidity of the structure. And this model gave us the theoretical expected volume of the organoid with respect to the 3 parameters. And as a second aspect of our collaboration, they also work on tumoral lung organoids. So it has been shown that most common pituitary tumors, in 2/3 of the case, are coming from breast or lung cancer. And this pathology is very difficult to diagnose and study. So our collaboration would result in connecting our 2 organoids of adenohypophysis and pulmonary tumor through a microfluidic device to construct an in vitro model to better understand how the metastasis invades the adenohypophysis coming from the lung. So in the end, we were very proud to be awarded the gold medal for our project delivered by the INOC organizers. And to conclude, we were able to develop an early adenohypophysis organoid from HIPS cells, expressing very specific markers, notably from pituitary placoid. We validate the protocol for the first 27 days of differentiation by selecting a very specific cell medium and a HIPS cell line. We were able to form a 3D retinal organoid expressing RP markers and rhodopsin, and we validate the first step of the organoid on chip formation. So it is very important to note that the success of our project relied on funds and communication, but also through partnerships with laboratories and suppliers, and also the scientific network. So our future work would be to adapt the adenohypophysis protocol to the microfluidic chip, and to improve the number of traps on the chip to be able to culture more organoids. So we would like to thank all the people we met during this project, all the researchers that gave us insights on the theoretical part, but also on the experimental part of our project, our supervisors Nathalie Lefort, Sébastien Sarr, Charles Barou, and Mata Pietri, and also all our sponsors and partners that made this project possible. Thank you for your attention.